Hello, welcome everybody to this last webinar of this cycle devoted to the role of complexity science in these new, let's say, settings that we are living together. And well, this is the last, and then we expect, so with this we will finish this cycle of webinars. But then we hope to be back later in. At some point after the summer break, and then we expect to have more webinars or hopefully seminars or whatever, and then being back and dealing with these special topics, and then to see exactly what complexity science can offer to these new problems that we are facing today. So now, very well, you are very welcome, and then it's a very pleasure to have today here with us Ernesto Estrada, and then I give my word to Roger Guimerà, who will introduce Ernesto to all of you. Please, Roger.
Okay, thank you, Albert. So, well, it is my pleasure to introduce Ernesto Estrada. I'm positive that many of you know him very well already, so whatever I'm going to say is not really necessary for him, but anyway, they pay me to do this, right? No, they don't pay me. But anyway, so since for the last year, more or less, since January 2019, he's the RAIT researcher at the Institute of Mathematics and Applications at the University of Zaragoza. As you know, his research interests are broadly in the area of discrete mathematical models for complex systems. You know that he's done lots of things on networks, on a bunch of other topics, right? Both theoretical and applied. And he's published over 200 papers and a couple of textbooks as well with Oxford University Press. And all these works have received in excess of 13,000 citations. So that's why I'm saying that everybody knows him, right? Because he's been very highly cited, and his work is having a huge impact on what all of us do, right? Or many of us do, at least. Okay, so he's also the editor-in-chief of the Journal of Complex Networks and associate editor of the Journal of Applied Mathematics from the Society for Industrial and Applied Mathematics, and of the Proceedings of the Royal Society A. Well, you've met him, talked to him, seen his talks in all sorts of major international conferences, from the Society of Industrial Mathematics annual meeting to complex networks and computational social science, all sorts of conferences. And also you've seen him probably in teaching in summer schools or in winter schools and all these sorts of events. As I said, he's worked on a bunch of topics that have affected how all of us do research, or many of us do research. But today he's going to be talking about fractional diffusion in the human proteome as an alternative explanation for the effects that SARS has on, or the new SARS coronavirus has on organ damage other than lungs, I think.
Thank you very much, Roger, for the nice introduction. And also, thank you very much, Albert, for inviting me to this webinar. I'm very happy to be here in Barcelona. Oh, no, that's not the case. So, but it is like being there, because I have very good remembrance from my last seminar and in your institute. So yeah, I will talk today about some very new results. Everything about the SARS CoV-2 is a new research. Before starting, I want to just show you that I have submitted for publication, is now on the review now. This review article about the COVID-19 and SARS-CoV-2 is mainly about everything which is related to modeling, from epidemiological analysis, all the compartmental and multi-compartmental models that has been used, but also non-compartmental models. The problem of predictability of these models is also discussed here. Then a second part about modeling for drug repurposing, and which there are many different areas here. I have classified this by the different proteins that people has been using for helping in this important task. And finally, about modeling for vaccine design, and in particular for epitopes vaccines, which I think are the most important and interesting one for a few characteristics that they have. So if you want, because it's not in the archive, so if you want a copy of these, please email me to estrada66@unisar.es, and I will send you a copy of the review, and hopefully will appear very soon in the journal. So I will start now by remembering what is the COVID-19 pandemic. So we know all very well, because somehow we are involved in this, we are part of this. And this started in December, or at least the most recognized date for starting is in December 2019. And in January 15, there were only very few cases reported, according to the World Health Organization in China. A month later, it has expanded to several countries, mainly in Eastern Asia, but also already in North America. And then a month later, you already have many of the different countries in the world has been hit by this outbreak. And in very few months later, we have practically the totality of the countries in the world that has been affected. So what is this disease? Well, it's a disease which is transmitted by mainly by direct and indirect contact. By indirect, many people means different things. For instance, in this very recent paper of Richard et al. In Nature Communication, they consider that it's transmitted from one person to another, just opposite to it. This is direct, and indirect is transmitted by the air. And they confirmed experimentally this by using ferrets. And but also indirect means that some of the droplets which are produced by cough of a sneeze of somebody which is having the disease can be too heavy and then drop to a surface. Somebody else can touch this surface and then can put their hands into their respiratory ways, and then these droplets can go inside by the nasopharynx system in the human body, and then it goes to the bronchi, and from goes to the lungs, and in the lungs it goes mainly to the alveoli, in which a pulmonary severe respiratory disease starts. So basically, we had the outbreak of 2002 and 2003, and this is produced by a relatively similar coronavirus, but we will see that there are some important differences between these two coronaviruses, and this is originally classified as a severe respiratory acute respiratory disease, but as we will see, there are more affections than this one. So this is produced by a virus which is named Severe Acute Respiratory Syndrome Coronavirus 2. This is for distinguishing from the coronavirus 1, that was the one producing the outbreak of 2002 and 2003. This is a microphotograph of the coronavirus. The most interesting or characteristic feature of this virus is these spikes that you can see here. These spikes are not other thing than the characteristic spike glycoprotein, which is a trimetic protein that is divided into two main parts, and one of these parts, which is the part that is recognized by the human cell, is just here in the top. And then the virus has another proteins, as we will see. It's an RNA virus. We can say that it is one of the biggest RNAs in virus that affect humans. It contains a membrane here, and there are some glycoproteins as well in the membrane. I will show you a little bit later some of the main or the most important proteins in the virus. So the life cycle of the SARS SARS-CoV-2 start with the so-called endocytosis. And this is a receptor-driven endocytosis in which these proteins, which are the spike proteins, are recognized by a human cell receptor, which is the ACE2 receptor. I will say AC2 receptor. And it is also necessary to have these another membrane protein here, as we will see later. And then the virus enter into the human cell and the cytosol and form this cytosome here. And at some point, this is merged with the cover of the envelope of the virus, and the virus liberates all the viral material into the human cell cytosol. And this is basically liberating the RNA and all the proteins that the virus has. So the RNA, the messenger RNA, is then translated by the ribosome of the human cell. So the virus sequester, hijack the ribosome of the human cell in order to translate this RNA into two long polyproteins. These two long polyproteins are produced in this translation mechanism, and then two proteins that has emerged from the viral interior here, which are two proteolytic proteins. The first is the papain-like protease, and the second one is the main protease of the SARS-CoV-2, produce the proteolysis of these two polyproteins and produce 16 non-structural proteins. So I have to clarify that in structural biology, non-structural means something different than in virology. So in virology, non-structural means not a disordered protein, but a protein which is not taking part on the envelope or the structure of the virus, like the spike protein or other proteins which are forming part of this envelope. So then there are several other parts here, the transcription of this complex formed by the RNA and some of the proteins, non-structural proteins here. And another important part is a second translation here, which occurs in the nucleus of the human cell. And in this particular case, the structural proteins are formed. So this goes to the Golgi apparatus of the human cell, and they are assembled there, and again is produced this lysosome here, and then the virus is again released by using an exocytosis kind of mechanism. So this is more or less the standard mechanism in which the virus is living. And as we can see later, this has an important implication in what we are going to talk today. So the first thing that we can find is that the clinical effects of the COVID-19 is not only constrained to the lungs. So it's not only a respiratory disease. There is an hyperinflammatory response of practically the whole body. And there are also involvement of the ACE2 enzyme in several organs, which can produce, for instance, heart failure, renal failure, liver damage, shock, and in general, multi-organ kind of damages. So you can see here, there has been neurological affections, there are heart problems, of course, the lung ones, there are problems of coagulation, forming of clots, and related disease with that. Also, it has been reported damage in the liver and the gut and kidney, but you will see that there are other potentially damaged organs as well. So one of the potential mechanisms for this happening, this multi-organ affection happening, is the so-called cytokine storm. And the cytokine storm is not any new thing of this virus. So the most dangerous case of influenza has been also reported to produce the cytokine storm, and this basically starts with the entrance of the virus into the cell, and then the immunological system of the cell start to respond, and then it generates cytokines and also chemokines, and these are pro-inflammatory molecules that will produce the inflammation of the organs in which they are liberated, and this goes to the blood flow here, and then this produces a sort of vicious cycle in which more cytokines and chemokines are generated, and then the inflammation of the corresponding organs produce damage in this particular organ. However, as I said before, it was very recently proved experimentally that SARS-CoV-2, in a similar way as the SARS-CoV-1, used the AC2 receptor for entering into the cell. So the AC2 receptor, which is the angiotensin converting enzyme 2, is used as the recognition of this particular S protein. The RBD part of this protein is recognized here, and then it is also used this transmembrane protein, which is the type 2 transmembrane serine protease, in order to produce the lysis of the S protein here. And what has been already determined is that the affinity of the S protein of SARS-CoV-2 for the AC2 receptor is significantly higher than the capacity that the SARS-CoV-1 had for recognizing this AC2. This is another problem as well for animal models, for instance, for biologists which are listening to this talk, is that, well, it's difficult to find the same kind of susceptibility of the virus to AC2 receptors in animal models. Of course, there are kind of biotechnological approaches in which this can be produced. But the important thing here is that this is a very important receptor for recognizing the virus and entering into the cell. But this receptor is very ubiquitous in the human organism and the human organs in general. And in particular, in certain kind of diseases, it has been found that AC2 is increasingly expressed. For instance, in the case of human heart diseases, AC2 has been with expression increased at both RNA and protein level. So then it means that this heart can be infected by, or this person, sorry, can be infected by the SARS-CoV-2. There is a high probability that this person can suffer from heart conditions aggravated by the interaction of the AC2 in the heart with the coronavirus. And this situation is not only for the case of the heart. This is a sort of review meta-analysis of the situation, and this author have found that this AC2 is ubiquitous in many different organs. And for instance, it can have a neurological manifestation. So the potential neurotropic mechanism of COVID-19 may also be due to the viral passage from the systemic to the cerebral circulation through the microcirculation, followed by binding to AC2 receptors in the capillary endothelium. And this happens for other organs as well. Here it can happen for the pancreas, it can happen for the esophagus, ileum, kidney, bladder. And here is a very recent finding that percentage of people affected in critical care by COVID-19 were suffering strokes. And here there is the hypothesis that the depletion of AC2 by SARS-CoV-2, because this receptor is then used by the spike protein, may tip the balance in favor of the AC1 angiotensin 2 excess that then can promote injuries like a stroke in these patients. This is another paper in which neurological diseases are also potentially related to the binding of AC2 in the central nervous system. So all of these findings are in favor of thinking about that AC2 and the abundance of AC2 in different organisms could be responsible for the multi-organ damage produced by the disease. But then if you search in the literature, we found an interesting paper 1 year later of the outbreak of the SARS-CoV-1. And this is in the Journal of Pathology. And Hamming and co-workers determined the abundance of the AC2 on endothelia and smooth muscle cells in many different organs in humans. And what they found is that this receptor is very highly expressed in most of the human endothelia and smooth muscle cells. And the lungs are not the ones having the largest expression of this receptor. So what this suggests, according to these authors, is that, of course, when the virus is circulating through the body in an infected person, then practically every single organ in the human can be affected. However, what it was found for SARS-CoV-1 and now for SARS-CoV-2 is that not all organs are affected, but some selectively target organs are the ones which are affected. The author wrote here in a very, very remarkable way that the absence, however, of SARS-CoV in these organs, it means in all the organs, as shown by in situ hybridization studies, is at variance, is against with this assumption that this AC2 is the major responsible for the multi-organ damage produced by this coronavirus. You know that there are important differences here between gender and between different ages. So you were listening to Alex Arenas in the previous seminar. Their model need to take into account different age strata, because different age are affected in different ways by the virus. And more importantly, most of the people that goes to intensive care having a very problematic consequences with the infection are in the elderly strata of age here. However, in this paper by Lee and co-workers, they found that AC2 is not really doesn't have really differences in its expression in different kind of genders, different kind of age, or even different kind of races. So they conclude in this paper that for organs different from lungs, because for lungs this difference exists, but for organs different from lungs, AC2 expression level have no significant difference between male and females, young and old person, and between Asian and non-Asian races in various human tissues. And this is experimentally determined. So this is against the idea that AC2 is at least the only one mechanism for this multi-organ damage. So when we were reading this kind of material, we were thinking, okay, is there any other possible route? And something that we sometimes forget is that when we have here that the virus envelope is fused with the lysosome, and all the viral particles goes to the human cytoplasm. In this human cell cytoplasm, there are proteins. So the virus liberates here 29 different proteins. So the virus contains only 29 proteins. 16 of them are non-structural proteins, and the rest 13 are structural proteins. All of them has been already identified, and also all of them has been detected to be interacting with certain kind of human protein. For instance, these non-structural protein 7 interact with all of these proteins in the human cell, including all of these proteins in this signaling way. This one, for instance, which is a non-structural protein as well, also interact with the DNA polymerase alpha complex and several others. You can see here the non-structural protein 13 interact with a huge number of proteins in the human cell, including the centrosome complex there. So although this is not the complete figure, so the figure has more interaction, but for the sake of simplicity, I will show you only this one. So what is important here is that these proteins, when they enter into the human cell, they are not doing nothing. They are simply interacting with other proteins. So if we now pick all of these proteins in the human cell, what we discover, and this was made by G.C. et al. In a paper in the archive by Barabashi's group, is that they identify that, for me, is very surprisingly, these human proteins are not randomly distributed in the human proteome. So they practically form a subgraph which is connected inside the human protein-protein interaction network. So there are few proteins which are not in the complex, but more than 90% of all the proteins in a human cell that interact with the viral proteins are forming a kind of connected network. And if there is a connected network, there is a necessary transmission of effect from one node of the network to another node of the network. So I am not taking here a technical definition. I suppose that in the previous seminars, people has defined what exactly a network is. But for the sake of this work, we are happy to say that a network is formed by these nodes, which are representing the proteins, and two of these proteins are connected if and only if they interact with exactly the same viral protein. But these proteins are inside the cell, and the cell is not like it looks in the books. So in the books, the cell, in the biology books, and I bought a couple of biology books recently, and the cell appears like a very empty place with the nucleus here and the cytoplasm and blah, blah, blah. But the cell is a very crowded environment. This is a pictorial representation from simulations of how macromolecules looks inside a cell. So it is a very crowded, it's a very packed environment. I have read that there are 42 million proteins inside one human cell. And this has immediately consequences for the way in which one interaction, one perturbation in one protein is transmitted to another in this protein-protein interaction network. So we cannot think that if we perturb one protein, it immediately transmits this perturbation to the nearest neighbors. This is completely unrealistic. So if you go to the literature, we can see that the main effect that this crowding environment has on the processes taking place on the cell is that there is no normal diffusion in which the mean square displacement scale linearly with time, but it scale as a power law with an exponent beta, which is smaller than 1. And this is what is known as subdiffusion. And there are different mechanisms that produce subdiffusion. And this is to show you the complexities of the problem that we are trying to target, is that if we try to simulate this interaction between two proteins that try to encounter one to the other inside this crowded environment, we can go to the class of viscoelastic diffusion, which is characterized by long-range, power-low correlated noise, by Gaussian displacement probability density. It means that they are ergodic processes. And typically, you can use the generalized Langevin equation with power-low friction kernel, or you can use a method which is very popular among physicists, which is the fractional Brownian motion. But then there is another category of models, which is the class of the continuous time random walk diffusion. In this particular case, we have a scale-free power laws waiting time in between motion events. So what it means that you perturb one protein here, and then there is a time that elapses until this perturbation is transmitted to the nearest neighbor. And these times elapsed from the perturbation of one protein to the transmission scale as a power law for the global picture inside the cell. So typically, they are characterized by displacement probability, which are not Gaussian. They are typically stretched Gaussian. So then it means that they are not ergodic. So there are breaking of the weak ergodicity principle. There are aging characteristics. And the fundamental equation here is the fractional Fokker-Planck equation. Then if there is no drift, or if you want to make a simplification, you remove the part of the Fokker-Planck fractional equation for the drift, and you got a fractional diffusion equation. Of course, this is theory, so the simulations will give you some insights in one case or the other, but what the experiments says? Well, this is a paper by, there are many papers by Matthias Weiss, and you can find many of them. This is in PRL 2009. They were studying experimentally the diffusion inside a cell by using a technique which is named a fluorescence correlation spectroscopy, and using simulations. In the simulations, they used the fractional Brownian motion, and they found that the fractional Brownian motion, or another technique, is a struct percolation like diffusion, reproduced very well the experiment. More importantly, they conclude that continuous time random walk with a non-Gaussian propagator is not able to reproduce their experiment of diffusion of a macromolecule, a protein in this particular case, a globular protein to be more precise, inside the cell. So these point out to that the real thing here is the class of viscoelastic diffusion. But then we found this another paper, and of course there are many other different papers in the same direction. And these authors use a state-of-the-art neutron spin echo and also small angle neutron scattering, also for the diffusion of a globular protein inside a crowded environment, in this case produced by a polymer. And they observe that the fractional diffusion equation was able to reproduce very well their experiments, and this belongs to the class of continuous time random walks. So we have a kind of contradiction here. But if you analyze what's happening, there is no contradiction at all. What happened is that the experimental techniques that they are using, and this is a very important advice to younger students, is that it's not necessarily the first thing that we see as experimental verification is all right. So we need to see what are the framework in which these experiments are done. So I'm a little bit pressed by time now, but here you can see that the fluorescence correlational spectroscopy works in the framework of 10⁻⁶ seconds and in the micrometers kind of length. And the other two techniques cover a very wide range from 10⁻⁻10 seconds to 10⁴ seconds, and a very wide range of length as well, from nanometers to tens of micrometers. So what justifies the difference finding between these two techniques is that they are observing completely different kind of scenarios. So here is a very nice review by Grimaldo about diffusion of proteins in cellular environments. And this diffusion is not as simple as we can think. First, at the very initial times, I could say that this is when the protein wakes up and say, I want to diffuse. There is a ballistic diffusion here. So the mean square displacement scales with the second power of the time. But this is a very, very short time. Indeed, I will ignore this part for this work here. But then it starts to a normal diffusion process because the protein is still not aware of the crowdedness of the environment. It is trapped in this hole here, and it can diffuse as a normal random walker. But then it goes to the scenario when the protein feels the crowdedness of the environment, and then you have here a subdiffusive process, which is taking place in the region of about 100 nanoseconds. And for the longer times, what the protein is seeing is just a very long steps inside of this completely crowded environment, which is equivalent to a viscoelastic media. So if you go like in the fluorescent correlation spectroscopy to very long time and relatively long length scale and relatively long time scales, what you see is not these holes here, but simply you are seeing a completely viscoelastic environment. So here is the global picture. So this paper is completely right, but is seeing the a little bit more macroscopic movement of the protein, while the combination of these two neutron scattering techniques take into account both the movement inside the traps and the movement in this viscoelastic media. And because this is modeled by the fractional diffusion equation, we are going to use a fractional diffusion equation in our work. So I necessarily have to go a little bit quickly here, but just the main concepts. So instead, simple derivative as the ones that we use in calculus, we use the first derivative of the function f, the second derivative, the third derivative, and so forth. Here we use fractional derivative. So here alpha is a number between 0 and 1. If we have 1, we have the first derivative here. And if we have a fraction here, we need to define this by using this integral. And in this integral, it's important that this delta here is the scaling function of alpha, which is this coefficient here. So suppose that we have alpha equal to 0.5, so delta is equal to 1, and then I will have here the the gamma function here, and I have this integral of the first derivative of the function evaluated in tau from 0 to t divided by t minus tau raised to alpha, because these two cancel for the case alpha is just 0.5. So it's a generalization of the case of the normal derivative. There are many different ways of defining this fractional derivative. For physical reasoning, the Caputo fractional derivative, which is defined here, is possibly the most appealing one. So here we use the typical Laplacian operator on a network. So this is an operator that takes any function on the vertices of the network and sum the difference between this function applied to nearest neighbors nodes. So this is just the normal Laplacian on the network, and this function could be any complex function defined on the vertices. And now we define our time fractional diffusion equation. And this is important because here the fractional part comes from the time. So know the space. So the space is remaining in the simplest way as we recognize for the diffusion equation. See, here is the diffusion coefficient that we are going to just put equal to 1. But this derivative in time with a fractional exponent captured this idea that the perturbation at one particular node need to wait for a power law time to be transmitted to the nearest neighbor. And as we will see, this is exactly what this is capturing. So we can prove analytically that this system always converge with any initial condition here. Of course, if the graph is connected, and we are working with non-directed networks. In case of directed networks, for full convergence, you have to have a strongly connected network. Otherwise, there is a convergence in each of the strongly connected component of the network. And here, the solution is given by this time fractional operator multiplied by the vector of the initial conditions. And this fractional time evolution operator is the so-called Mittag-Leffler function. And a Mittag-Leffler function is an extension of what we have seen, for instance, for the exponential. Indeed, we can take here the Taylor series in which the infinite sum of all the powers of this metric m divided by a generalization of the factorial, which is the gamma function. And in this case, we have the gamma of alpha, which is our parameter here, 1, because our solution always depends on alpha, but the second parameter in the Mittag-Leffler function, typically taken as beta, is fixed at 1. So you cannot move this parameter because then you are moving out of the solution of this equation here. And this is just defined in this way. So you possibly know that the Mittag-Leffler function for alpha equal to 1 is just the exponential. So then it means that for alpha equal to 1, this equation here is the simple diffusion equation for a graph. So then it means that this is a generalization of this equation. But if you have, for instance, a fraction here, you can have the exponential of the second power of the metric, the identity matrix minus the error function of the metric n, and so forth. So I think I will skip this part here, and then I will go to these experiments. So first, we were trying to understand how this system works in the case of our protein-protein interaction network. And then I will just put this symbol here to indicate that I will start the process of perturbation at this particular protein. This is just for initial experiments. In the final experiments, we started the process by perturbing the protein that interact with the S protein. That is supposed to be the first encounter of the human cell with the viral protein. But in order to see, we take this protein which is highly connected. It's a hub in this particular network. And then what we see, typical characteristic of subdiffusion, when the exponent here of the time fractional derivative is smaller than 1. So for alpha equal to 1, we have the typical Gaussian behavior, and this is the distance from the initiator of the perturbation. So in reality, this is just the from 0 to 10, but for the sake of a better picture, I symmetrize the plot in order to obtain this Gaussian-like shape. But then what happened is that, as you can see, when alpha is 0.5, at relatively long distance from the initial point, you see that this perturbation is propagated in a slower way. If we make a zoom of this, we get the following interesting picture. For very short distances from the origin, the subdiffusion is faster than the normal diffusion. But then for longer distances, it is slower than the normal diffusion. So we have a very fast initial stage for the nearest neighbor around the initial point, and then you see that this rate decrease dramatically with the distance. Because this is just one of the hubs of this network, we took a protein here with just degree 2, only connected to 2 other proteins, and what we see is exactly the same. The order of magnitude is different, but here you have that the subdiffusive process is faster at shorter distance from the perturbation and slower at longer distance from the perturbation site. And this is exactly what we want to reproduce. So we want that case in which the protein field that is inside a trap and has not filled yet the trap, the crowded environment in which it is embedded. Now we are going to see what happened with time. And here I will compare for the time fractional derivative for alpha equal 1, so the normal diffusion, and the time fractional diffusion for alpha equal to 0.5. So if you see at long times, this process, the normal diffusion process, is converging much faster than this fractional or subdiffusive process. So at long time, as expected, you have that this normal diffusion is faster than the subdiffusion. But if you check here at short times, so making a zoom here at this region, we see that the concentration or the probability of being perturbed by the original protein here increase very quickly, like in the shock wave with the initial propagation. And what happened is that this shock wave appear much faster in the subdiffusive process than in the normal diffusion one. So see here that some of them appear almost immediately after the perturbation is applied to this hub here. And this is taking into account all the proteins in the protein-protein interaction network. What happened is that this process, the fractional time diffusion, is fast. So the subdiffusion is fast at shorter time, but very slow in the long time in general. Then what we think is, okay, maybe we can not consider a single diffusion process here in cell, but a sort of switch and restart diffusive mechanism. So you start the diffusion at one protein here. There are some proteins which are highly perturbed. Then the process switch to this protein here and restart again. Because the time passing from this diffusion here to this diffusion here, we don't know, but possibly is very long, because possibly this protein is in one cell and this protein is in another cell. So you need to go to detect this other protein in a long way. So then, now this protein start the perturbation, perturb a few other proteins, and then these other few proteins, the process is switch these proteins and start again. So if we do this, but separating here the proteins in blue, which are mainly expressed in the lungs, and the proteins in green, which are mainly expressed outside the lungs, and we know in which organs are they expressed, what we observe is very similar. So now we are perturbing one protein which is in the lungs, and we observe what happened in the case of the normal diffusion for the proteins which are outside the lungs. And what happened, if you compare this for alpha equal to 1 with alpha equal to 0.75, is that at the long time, the first is faster. You can see here, there is almost a convergence to the steady state. This is far from the steady state. But at short time, the second one is faster than the previous one. see here that this shock wave occurs very early in the initial stage of the perturbation of the protein, in this particular case, GOTA2. Now, what we did is just to consider how much perturbed these proteins outside the lungs are if you perturb one by one each of the proteins which are in the lung. Okay, so I pick one protein, I make a perturbation, and then I count how many of the proteins outside the lungs are perturbed. What is the probability that these proteins outside the lungs are perturbed, and the time at which this peak occurs in these proteins outside the lungs? And this is what we found. So we have, this is the top ranking here. So PKP2 is perturbed by 21 different proteins in the lungs. And where PKP2 is mainly expressed? Heart. CEP43 is perturbed by 18 proteins. This is the sum of the probabilities here, and this is the mean for the perturbation process. Where is this protein mainly expressed? Testis. CEP135 in skeletal muscle, heart muscle, cerebral cortex, cerebellum. For instance, here it appears the thymus in several proteins which are perturbing it, testis in many of these proteins, and also prostate in several proteins. Particularly the prostate and the testis are typical only for male, and thymus is important for differentiating between children and adults, because the thymus start to be degraded with age at a rate very, very fast after we pass the 40s and 50s. So it has been hypothesized that affection of the thymus is very important to explain the difference between children and adults. And then if you search for the literature, you find evidence that COVID has been affecting the heart, genital tract, kidney, difference between children and adults, liver, testis, hypothalamus, pituitary, adrenal axis, etc. So finally, what we did is, okay, now we know what are the proteins that produce or which are more perturbed by the proteins inside the lungs. But what are the proteins inside the lungs that perturb more the proteins outside the lungs? So we can say that these are the disease activators, or the multi-organ disease activators, to be more precise. So these are the proteins that if we perturb them, the proteins in the other organs outside the lungs felt in a more drastic way this perturbation. So this is the capacity of a protein mainly expressed in the lungs of perturbing proteins which are mainly expressed outside the lungs. And once we have all of these results, well, we were relatively happy because the model was working okay. And then looking for reference for a completely different matter, we found this paper, which is in the archive. Postmortem tissue proteomics reveals the pathogenesis of multi-organ injuries of COVID-19. So these authors took different patients. From the patient, they took different organs. And from these organs, they made an analysis of the proteins which are either down-regulated or up-regulated post-mortem when these individuals were suffering from COVID-19. What we have found is that 58.1 of the disease activators that we reported previously were found by these authors to be down-regulated in post-mortem analysis of the tissues of these patients. But from the proteins which are not in our list of priority, only 29% of them were found to be downregulated in the postmortem analysis of these individuals. We have to add that they used only 110 of the proteins in the PPI that we use that contains 209 proteins. So they are working with half the proteome. And even in that case, these numbers, I think, are very interesting as a potential validation of our results. Also, they found, for instance, we found that these proteins that were predicted to be affected in different organs by our model were also found by these authors in the postmortem analysis. Here, PKP2 coincide, the heart and the heart. They didn't analyze testes, so we predict here testes. They found brain, no jokes about connection between testes and brain. RIP6 coincide, the liver and the liver. HOOK1, we predicted liver and testes, and they found the liver, heart, and lungs. And this one we found in the heart skeletal muscle that they didn't test skeletal muscle, and brain, and they found in the heart, so there is a coincidence here. So far, they analyze the lungs, kidney, liver, intestine, brain, and heart. And so far, I think this is a good validation for the model. Of course, it is a model and can be improved. One way of improving this is to use what we call, what we have named a metaplex. In the metaplex, what we combine is one continuous space inside each of the nodes of a network. And this continuous space simply can be modeled by a set of locally compact metric spaces with a given kind of measure. And then you can put here one diffusion equation, could be a fractional diffusion equation, that contains not only fractional time diffusion, but also space fractional diffusion, Levy flights. So we can allow here inside this cell not only a normal random walk, not only a subdiffusive random walk, but also a super diffusive random walker. And in this particular case, we can be analyzing these very initial tiny steps in the modeling. And between one node and the other node, we simply use the same kind of fractional diffusion to which we can also incorporate long-range interactions, but controlled by the Laplacian metric in a similar way as we have done here. So the main thing in the next addition to here is to detail more what could be happening in the cell for a given protein before this protein encounters another protein. So in conclusion, we have seen here that a time fractional diffusion equation with Caputo derivative and the switch and restart processes can be suitable for modeling the propagation of perturbation of proteins in crowded intracellular spaces, and the propagation of perturbation across PPI networks seems to be a plausible mechanism for explaining the multi-organ damage suffered by patients of COVID-19. And this is everything I have. So thank you very much for your attention.
Thank you very much, Ernesto. I think it was a very clear, great talk. So thank you very much again. Now, if there are questions, there were no clarification questions. Again, I am assuming that it's because everything, or at least to me, it seemed very clear. So now it's time to ask more regular discussion questions. Anyone? Because there are, there are the technical issues. I'll give a few seconds for people to decide. Okay, well, I have a question. I can start while people figure out how to do this, maybe, right? So I have a question about the difference between the connection between the abstract diffusion that you have in your protein-protein interaction network, right? And the physical diffusion that you have, let's say, between organs, right? So I understand that that the abstract diffusion in the cell, it's modeling, I don't know, this whatever protein that it's altered somehow by the virus then, and it's moving inside the cell, and it suddenly interacts with one of its interactors, right, with another protein that it's also expressed in a different organ. But now we are talking of a particular cell, right? You are diffusing within your cell in this very crowded environment, and then you interact with someone and you affect that some other protein, and that's the abstract diffusion in the protein interaction network, right? I am within one cell and I'm interacting with proteins with which I typically interact, and I affect them, and I'm propagating this perturbation within the cell. But now, how do you connect that to the fact that if I'm a cell that is affected in the lung, and I want to affect the heart, let's say, I don't know, I have to travel physically from one organ to the other, right? Maybe I have to get in the bloodstream and go from one to the other. Or so yeah. I was curious about how you see the connection between these two, let's say, abstract physical vision of the diffusion process, which I'm not sure is very relevant. But
and we really thought a lot about this and found also the fact that proteins emigrate from inside the cells and navigate outside the cell to find other cells in other organs. That's possible. This is one of the possibilities, and this is why we are using this switch and restart process, because we are not considering that the process is just immediately one after the other. So one protein, as you say, inside the cell is perturbed, it navigates inside the cell, and then it perturbs another protein in the same cell. But then this protein can navigate out the cell and then can be interacting with another protein in a different scenario. So this is one potential scenario. The other one is that we really are considering here that the virus is at a concentration that allows it circulating across the whole body. So it's exactly the same as the situation when we are thinking for the ACE2 receptor, and then the virus is at a concentration in which it can interact with a protein in one organ or in another organ. So in reality, the connection is still something that we have to work more. And I think the part in which we can think that the protein goes out one particular cell and can be interacting with another protein in another cell, for which, to be honest, not too much experimental evidence, but some of them has been reported. I think is the most interesting picture for the kind of mechanism that we are proposing, which is this switch and restart kind of.
Okay, thank you very much. So now, Johan finally, asked the question.
The question is the one that I recently wrote. Did you define the alpha exponent as a continuum exponent, between 0 and y? Why did you compare only with alpha exponent above 0.1? Why not 0.2 or why not 0?
Okay, that's a very interesting question. We really explore what happened for different values of alpha. And what happened is the following. If you observe already in the figures for alpha 0.5, the increase of the perturbation of so you have a protein here, and you have several proteins. If you protein here, which is perturbed, and this protein will be felt by other proteins which are nearby that protein. So we have seen that the probability that this neighborhood of proteins be perturbed increase very quickly in time. What happened is that the smaller the value of alpha, the fastest is the increase in this probability of perturbation. So for value of alpha equal to 0.5, this increase in the perturbation is instantaneously. So it means that you are perturbing one protein and instantaneously the other proteins are feeling the perturbation. So of course, even for a quantum world, that could be a very weird situation. So we try to avoid that, and that's why we select a relatively large value of alpha, which is alpha 0.75, because at 0.75, you still have that the time at which this perturbation probability increase is faster than for the normal perturbation, but it's not instantaneous. So the situation is, I can show you in the slides, but the situation is that that for values of alpha very small, and very small means in this case below 0.5, you have an instantaneous increase in the perturbation probability of the nearest neighbors of a perturbed protein, which is not realistic. And we try to avoid this by moving to bigger values. But in reality, we tested computationally in the simulations many different values of alpha across the interval between 0 and 1.
Okay, thank you very much. And maybe I'm not sure if I have another moment for a silly question. That is,
you can do it. Go
okay,
ahead.
Thank you. Maybe it's a naive question. If the sense of a non-fractional derivative is related to, I don't know, change rate or velocity, what could be the meaning of a fractional derivative in a physical way?
Okay, so you can go to a paper by two experts. Minardi is one of the authors, and it was one of the most well-known experts in the mathematics of fractional calculus. And there is a paper attempting to explain what is the physical meaning of the fractional derivative. To be honest, I was not very happy with the explanation because it seems to be a little bit of fantasy. Okay, so he imagined two different kind of times and try to relate one to the physical time, another not. But what you can think about is that the fractional time derivative try to account for this kind of time delay that you have in the propagation of one effect from one particular event to the next one. So it's not exactly to have a waiting time, because this you can put a waiting time which is scaling as a power law, and in this particular case, I completely sure you will end up with a kind of fractional derivative. So the simplest meaning for the fractional derivative is that it tries to capture this power law waiting time from the perturbation of one particular event to the next one. But if you want the paper of Minardi and I think it's Gorenflo, the other author, they explain in a wider way what the fractional derivative is. By the way, fractional calculus is not new. It's a little bit later after Newton and Leibniz. Already mathematician asked, well, if I have the second derivative, why not the 1/2 derivative? And from that point, it started to be working. But the most important part of fractional calculus was when the physicists started to use it in physical processes, and in particular in these scenarios where self-diffusive of any of the two kinds I show you today are observed in the physical world.
Okay, thank you very much, Ernesto.
You're welcome.
Okay, so are there any other questions?
I have one myself.
Oh,
yes, because I am the moderator, so I can do it at any time. Ernesto, you mentioned, the question is also kind of a formal like the last one, because you mentioned at some point this of a metaplex. So actually you said that the metaplex is like when you have a network and then in every single node you have a continuous description of what's going on. So it's a kind of what we usually talk about, a network of networks and so, because then we have a kind of hierarchical organization. But then this is what you have in mind. So this is the proper description of the system in this way. So you can define the analogous, for instance, of a Laplacian matrix, also in this case of derivatives and so in this construction.
Well, it is more general than a network of networks, because inside the nodes, you can have a network or you can have a continuous space. And in general, we are using a continuous space because otherwise you are in the network of networks. And then you can have certain boundary conditions in these nodes. So for instance, you can have one source from which a protein can enter into this particular node, and a sink from which this particular protein can go out from this particular node. And then you can also analyze the size of this sink and source. You can analyze the proximity of this sink and source. Even you can put then that they coincide or not, and so forth. So why I think that this is a better picture? Because we, in the paper which is now in press after 2 years in Science Review, we analyze a couple of metaplexes. One is in the brain, and the other was a landscape metaplexes in which species are moving and so forth. And what we observed is that there is a very different behavior in the case of the brain, depending on the characteristic of the topological network that you have and on the kind of dynamic that you have inside the nodes. Of course, the kind of dynamic that you have inside the nodes could be of any kind. It could be diffusion, reaction diffusion, it could be synchronization, whatever you like. But even for the simplest diffusion, what we have found is that in some cases, the dynamic inside the nodes determine the global dynamics. Saying in other words, the network plays no role. In other cases, what happened is that the network, the dynamic on the network determine the global dynamics. Saying in other words, there is a little influence of what is happening inside the nodes. So the two extremes are two extreme cases, of course, one for a very small world and the second for a very large world. But then we have all the intermediate scenarios which are describing more realistic situations. Like, I think the one that we are analyzing today, because of course, yeah, well, I am considering that the encounter between the two nodes occurs immediately in the way between the edge of the graph. But what happens if you take a metaplex is that inside the node, the protein will have to make a navigation to find the sink, going out the sink, enter into the source, do another random walk, and then found the protein. And this is exactly what happened in reality. So there are several papers when the experimenters try to analyze the encounter between two proteins in a cellular scenario, and it's very much like this. So it could be that the situation is not very different from what we have observed right now, and we already know or have some hints that when these cases can happen. But it could be the case as well that the situations are different.
Okay, thank you very much.
So in the meantime, there was another question in the chat, which I'm going to read. Claudio Rocco. He says, nice presentation. I have no microphone access. That's why I'm reading the question. So during the diffusion, what is the body doing? Just watching what happens, or there is a protective mechanism? Thanks.
Oh, wow, that's a complicated stuff, of course. So,
for a physicist, at least, right?
I suppose that there are many things happening at the same time, and of course, some of these proteins which are inside the virus, which are non-structural proteins, are known to have the function of trying to knock out the immune system of the human cell. So they will be doing this kind of job, and that there could be a response for that. And on top of that, there could be all the cytokine storm, and on top of that, it could be the navigation of the virus and interacting with other kind of cells by using the AC2 of those cells, etc., etc. So what we have done here is a model, try to simplify all these scenarios in order to try to give another potential route in which this multi-organ effect could happen. And why is this important? Think about the following. If the multi-organ effect were only produced by ACE2, or sorry, by the cytokine storm, then if you use anti-inflammatories, like steroid anti-inflammatory drugs, you will solve the problem. But we already know that this doesn't solve the problem at all. It partially solves the problem, but it's not the whole solution. If all multi-organ damage were produced only by the interaction with the AC2, then if you suppress the AC2 in certain cells, you can solve the situation. However, it has been shown that at least in experimental animals, this doesn't solve completely the problem. And some patients, like having heart problems or blood pressure problems, this could be a big problem. So if this mechanism that we are proposing here could be a plausible one, then using some drugs that intersect the interaction of some of these proteins at these organs, possibly, as in the paper of GC et al., they show there are already drugs that target some of these proteins, and using these could protect organs against this multi-organ damage. So it's, of course, a simplification, as any model do, but we would like to increase the complexity of the model in the future. Thank you.
Okay, Ernesto. Thank you so much for the talk and for the discussion, which I think has been very illuminating as well. As you know, this was the last talk in the seminar series. So I don't know if Albert wants to say some closing, make some closing remarks.
Well, I just would like to thank Ernesto for having this. It was a very nice talk. I think the idea precisely is that we are in a community of very interdisciplinary. So that's true that we are mainly physicists, some mathematicians, and but really that we want to know which are the real problems of where we can make. Let's say we can apply all our knowledge to these fields. And actually, I had the feeling during your talk that it was sometimes I had the feeling to being the talk was by a biologist, sometimes by a physicist, because you were looking so familiar with all these different terms that at the end you get okay. That you be someone that then closes this cycle and then. Really using very serious tools from mathematics, from physics, solving these, or at least trying to solve these very serious health problems which affect to all of us. And okay, just thank you and thank all the speakers of these webinars. I think it has been a great experience for all of us. And I will just close this. We are mid of July. We are usually facing the final of the course of the season. So looking for the summer break, that this year will be special, different for all of us. It will be far from normal. And okay, just looking forward to see all you people back in September, perhaps for more webinars, perhaps for more discussion, what people from complex system science can do for real problems like those that we are facing today. Thank you very much to all of you, and have a nice summer, and please be safe. Bye-bye.